Okay, so recapping on last week. Last week we covered a bunch of different equipment that we use in iGEM. Some of that is autoclaves, microcentrifuges, thermocyclers, nanodrops, gel electrophoresis chambers, and also transilluminators. I included pictures of each of these things here. This is an autoclave. It's also called pretty much just the sterilization chamber. It runs at around 15 psi at 121 degrees Celsius, and you use this to either sterilize substances such as equipment or broth, or just to mix together and get it, properly solubilized within the broth mixture. So you have like gel that you want to dissolve into it. Microcentrifuges are pictured here. These are just used to spin down tube contents. There are much larger centrifuges. The microcentrifuge specializes in particular size epis that you use for these. Basically what happens is, depending on the density, the size, shape, and viscosity of whatever substances that you have within the tube, those substances can begin separating, usually via whichever one's the largest variation. Thermocyclers, as pictured here, they're a pretty simple machine, though the GUI is kind of complex. It just varies temperature with time. It has a bunch of slots where you can put PCR tubes in there. And it does it often in repetitions or cycles. So it does 50 degrees Celsius for 20 minutes, 10 times, or et cetera. It can change the temp in between those cycles if you really need to. These are predominantly used for polymerase chain reaction, which we'll hit a little bit later in this lecture. Also, I hit it in the past. Then nanodrops, we used this last week. This is just a device. It's a spectrophotometer. I can't pronounce it. I'm telling you, man. This is used to measure DNA concentrations. And what it does is it shines certain wavelengths of light into the sample, and you can thereby extrapolate what concentrations of whatever DNA or RNA or proteins that you have within your sample. Finally, gel chambers and transilluminators, these are kind of used hand in hand. Gel chambers are used to load your gel electrophoresis gels. You pour the liquid gel into it, let it solidify. You load your either plasmid or restriction digest samples into the wells. You run it for an extended period of time, and it separates your DNA bands. And then the transilluminator is just a UV light that allows you to see those fluorescent dyes and take pictures of them when you're running a gel. So why Gibson Assembly? Previously, I covered standard and 3A assembly. Standard assembly was used way a long time ago, probably over 30 years ago, for transforming and editing genetic material for typically bacteria. And 3A assembly is kind of a further on version of that, where you're using different antibiotics in order to separate your plasmids when you're screening for them. So there are a few disadvantages to each of these types of assembly methods. The standard assembly has a lot more than just this with regards to disadvantages, but a few include you can only insert a single gene into a single plasmid at a time. Unlike 3A assembly, which you can do 2 inserts at a time and put one ahead of the other and then put that into an insert backbone. Standard assembly also, it requires gel purification because due to the lack of antibiotics, because you can't screen for it properly. And so you have to run it in a gel, separate your DNA's fragments in order to find the fragment that you need, and then purify that fragment so you can use it for other things. So it's I guess at that point it wouldn't be a fragment, right? It'd be a ligated, fully functional plasmid. But it's a long process, and you don't have to do that in 3A assembly. Also, standard assembly has a pretty, at least compared to these two, a lower success rate due to multiple factors. But one of them is having to gel purification, do that whole step, which decreases the efficiency because you're going to lose some, of course, when you do that step. As for 3A assembly, for the longest time, having 2 inserts was pretty good. But we've since come up with better methods of assembly that allow us to do up to 6. CRISPR allows you to do some crazy stuff. So we're a little bit past 2 inserts at this point, and so that I include is a disadvantage. Also, this makes it a little bit more complicated. So when you're doing 3A assembly, you have to have 3 different parts that have 3 different antibiotic resistance genes. And that could be if you don't have that in stock already, you're going to have to insert somehow those antibiotic resistance genes so you can actually use it in 3A assembly. So that adds some complexity to it. And then finally, blunt ends. So with 3A assembly, you can't really use blunt ends. You have to have overlapping restriction enzyme sites so they can connect to each other. So it's really hard to have blunt DNA that doesn't have restriction sites on the end already to actually use that for 3A assembly. Which brings me to Gibson assembly. This was discovered, formulated in 2009, so it's a pretty new procedure. It became very, very popular after Gibson and his associates published it. And it's a little bit different, but similar in a way to these first two assembly techniques. The advantages to Gibson assembly are, one, you can insert or ligate together up to 6 different fragments, which is 3 times the amount as 3A assembly, and it just makes this look shameful. It's also blunt end proof. So even if you have fragments that have blunt ends, meaning they don't have restriction enzyme sites that you can cut at the end, you can still use them for Gibson assembly. You can use them to ligate all the different fragments together. And this is because you can add actually restriction sites, though they're not required to do this blunt end proof stuff. You can add restriction sites as you want and whatever one you want to this using the Gibson assembly protocol. And finally, both of these require a plasmid backbone where you can insert your stuff. Here you can actually make a circular plasmid out of a few different fragments. So you can turn a fully linearized set of random fragments into a full circular plasmid. That's something you couldn't really do here. As for disadvantages, we'll get into those in the next few slides, but one of those is complexity in primers. So you have to do a bunch of pre-prep and research beforehand to figure out what primers you need in order to get these parts beginning to connect and ligated together. And also very expensive considering that you have to order those primers after you've designed them from a synthesization company. Yeah, so that's expensive. It's a lot better than it used to be, but it's still expensive compared to this. So this diagram helped me a lot with understanding what Gibson assembly is and how it works. This is actually only one of the techniques you can use with Gibson assembly. It's turning 3 different separate parts into a fully cylindrical part. And so like 3A assembly and standard assembly, the goal of Gibson assembly is to ligate a sequence of parts together or insert that sequence of parts into a plasmid backbone. And I included 2 here. You can either, like in this picture, you can take a bunch of linear fragments and turn them into a circular plasmid, or you can just say that you just have 1 or 2 of these, and you're going to want to insert these into a plasmid backbone, and that's what I mean by insert. So as for the sequence of doing this right here, or just Gibson assembly in general, you first start by adding the primers that you designed to the parts. So when you first start, these parts are not going to have these primers at the ends. And so you want to add these primers so that they match up. I'll get to that a little bit later. So that they match up and you can actually use them. So the first step is adding these primers. The second step is pretty simple. You just take these parts with their fully functional primers and you throw them into the Gibson assembly master mix. You do this in an epi tube, and you throw it all together. You're going to, of course, calculate the amount of fragments that you need via concentrations. So you can use the nano drop for that, or just PCR or something like that. But there is some calculation involved with just throwing stuff into the tube. Keep that in mind. After they've been thrown into the tube, part of the Gibson assembly master mix includes the T5 exonuclease. And what this does is it degrades the 5' end of the DNA fragment. And so what it does is it attaches here. This is the 5', this is 3'. So 5' is at the top, and it starts eating or degrading from the 5' to the 3' direction. So it just keeps on going down this and eating it and making pretty much like a hole there. And it does that for each one of these. So this is 5'. This, because it's reverse, this is 5', so it's going to eat this top one. It's going to eat this bottom one, this top one, this bottom one. And as you can see, now that we're eating this primer and this bottom primer are complementary, so they can start attaching together. And that's the gist of what's happening in Gibson Assembly. I guess I'll cover this a little bit later, but so yeah, after the exonuclease starts digesting the ends of the fragments. So as you can see here, it didn't show one of the colors, but it ate that. They start matching up, and they can start forming. So after they attach together via kinematics and just touching each other, and the complementary nucleotides match up, the TAC also included in the master mix is something called TAC DNA ligase, and this is what you use to start. Oh, actually, I missed something. Yeah, this step is the polymerase step. So after they attach together, that exonuclease is going to not just eat the primer; it's going to keep on eating it. So there's going to be, even if the primers match up perfectly, there's probably like a little segment here that's going to be empty. And so what this polymerase is used for, which is also in the mix, is it extends that primer so it fills in that gap, that hole that was made by the exonuclease, if it happens. Then finally, the last step, the TAC DNA ligase begins binding to this big strand, and what it does is it can finally form those covalent bonds between the phosphodiester backbones. Is that finalizes the process. You now have a fully ligated part that you can use. So I just want to briefly go back to this picture. This is an example of using 3 different parts in order to make a circular plasmid. And you can tell that this is the case because the blues go to the blues, the red goes to the reds, but then there's a green on each end. When you throw these into the mix, these greens are going to match up and form that circle. Okay, so this is a bit of a slide, but I wanted to briefly go into digesting Gibson Assembly primers. It's, I would say, the most complicated process of doing this whole thing. And because it's so important, you really have to put a lot of time and effort into designing these things. So before mixing everything together in the tube, you're going to want to start designing these primers. And you're going to use these primers in your PCR reaction in order to attach them to the fragments. I included, for your viewing pleasure, a video, I think it's from Khan Academy, that goes over PCR if you've forgotten what it was. I think they briefly mentioned primers. They don't go them into very much detail, though. It's mainly just the PCR reaction. I also, if you have questions about primers, I included a link here. I can't remember who created that video, but it goes into primers in pretty high detail. So if you have any questions about primers, click that video. So as I said before, there are different methods of using Gibson Assembly, inserting and making a circular plasmid, or maybe inserting multiple parts at the same time. So if you have a single fragment insert, when you're designing your plasmids, and by single fragment insert, I just mean you have one fragment and you want to insert it into a backbone. When you're designing your primers, you're going to design your primers so that the forward and reverse primers correspond to both the insert and the backbone. You can do this in 2 ways. I mentioned this in the tips. You could either split it half, so half the forward primer covers the backbone, and half the forward primer covers the insert, and then the reverse primer is the same thing. So it's half-half. It's kind of split down the middle with regards to the length of the primer. Or the other alternative is to not split it in half, but rather use the forward primer as just corresponds to the insert, and then the reverse primer just corresponds to the backbone. So that's the other way of doing it. Though I have more experience with the half-half, where you split it down the middle. And that's what this video down at the very bottom, it goes into. It actually was made by an iGEM team, I think Oxford. They made a very, very good video. It's more of a visual learning video where it goes into designing these primers so that it splits down the middle. Another tip when you're designing these primers is to end always when you're writing out your primers in whatever Word or a text file that you're working with, end it with cytosine and guanine, because they are much more stable compared to adenine and thymine. And this is the case because they have 3 hydrogen bonds. And so if you end it on that, it's going to be more stable than just having 2 hydrogen bonds that holds that end together. So yeah, always keep that in mind. Primers should be around 30 base pairs in length. So when you're designing this insert, you can do, or 60 base pairs in length, you're going to go 30 this way, 30 this way, 30 that way, 30 this way. So 30 are going to correspond to the backbone, 30 are going to correspond to the insert, and likewise for the forward and reverse. Of course, if you're just doing it like I said before, where it's the insert, the forward just does the insert, and the reverse just does the backbone, then it's going to be a full 60. Though that's not it's not a hard cutoff. You can be rough, say if you're at 60 and that ends on an adenine and thymine, you can move forward or backward, change that to a guanine and cytosine. Also, I didn't mention this, and it's a little bit too in detail for what I'm discussing, but if you're looking at the DNA sequence, and your primer ends at a cytosine and guanine, but right next to it is another cytosine and guanine, you want to move down one. So if basically having two cytosine and guanines together makes it less stable, is what I'm saying. As for the multiple fragment insert, so this is kind of similar to the single fragment insert, but instead of just inserting one fragment, you're inserting two. And so that is detailed here, where you have, or two or more, right? So you have multiple different fragments here. Each one corresponds and can start forming a big chain once they start being thrown into the master mix. And then these orange segments, these orange primers, correspond to the primers on the backbone. So the only thing that kind of differentiates this between the single fragment insert is the fact that you have to worry about these intermediary primers and make sure that the sequence of fragments is correct via the primers that you have. So say, for example, you have a promoter and you want that in front of SGFP. Of course, you want the primers to correspond to that sequence that you wanted. And finally, the last step, I kind of covered it on this slide, but it is the multi-fragment without the insert. So you're turning a sequence of fragments into a circular, basically plasmid. And that's detailed here. They match up together. I detailed that on the last slide, and they form the full circle. Yeah, that is all I have for this slide. Regardless if you don't watch these videos, I highly recommend watching that video at the bottom, especially when it comes to designing primers. Oxford did a really good job with explaining this in a visual manner, and the best way to learn this is through the visual, looking at the sequences, seeing what I mean by going 30 forward and 30 backwards. Or if you want to insert a restriction site, it's going to make it a little bit more complicated. So you're going to have your backbone, you're going to have your insert, and then you want a restriction site right here. So you have to take that into account when you're designing those primers. So that's pretty much it for the complexity of Gibson Assembly. I'm just going to finalize it with the protocol itself. It's 3 or 4 steps, depending on if you consider it putting in the freezer a step. The first step is just to gather the primer parts with the Gibson Assembly master mix and millicube water. All this is detailed here. The recipe changes depending on how many fragments you're doing, so keep that in mind. When you're designing what your volumes are of your fragments, you want to be very extra careful that it's unimolar. So you don't want to have more fragment than another fragment, because that will make the reaction a little bit finicky. And so for doing these calculations, I'm not going to go into detail here, but the NEB website, which I linked here, goes into figuring out how to find out how many volumes or how many grams or whatever, how many parts of fragments that you need for your mixture. So there's that. Next, you get all your parts. Make sure that you have the correct volume. So follow these for Gibson Assembly Master Mix, and make sure it's 2 times. And the deionized water, so millicube water. And you add all these to the tube, and you make sure it's on ice when you're adding everything, so it doesn't start the reaction before you want to. So you add them on ice, and then you take them off ice, add them to the thermocycler. The more parts you're using, the more fragments that you're using, the more time it's going to take. 50 degrees Celsius for 15 minutes if you have 2 to 3. 60 minutes if you have 4 to 6 fragments, those at the same temperature. You just incubate those, and after you're done, you put that tube into the freezer. Or you can use it in a protocol if you need. Here are my few external resources. I included a link on primer design, and then that's NEB's guide specifically on primer design. You can either do this or watch the video I posted earlier. And then for NEB's assembly protocol, which is also, this is the same link as here, this just goes over how you do Gibson assembly, what materials you need to do it, how long you should be doing it. So that's that link. And then a while back, I think it was 2010, 2011, Cambridge, they did because this was right when Gibson assembly was pretty new. iGEM Cambridge did an experiment with Gibson assembly just to test if it was useful for iGEM. Although Gibson assembly is not like pushed by iGEM, it's not like a certified assembly protocol. It is very useful and many teams use it. So iGEM Cambridge characterized it. So they have a bunch of tools on their website that you can click on. You can read some more information about Gibson assembly there as well. It's where I learned most of the material that I covered.